my name is Patricia, and I will present you my master thesis on the trophic ecology of shallow water black corals from the southwestern coast of Madagascar. Black corals are colonial organisms that belong to the subclass Hexacoralia order Antipataria. Unlike their closely related scleractinians, they don't build reef structures, but they can be found forming dense black coral beds, like the one in this picture. They present a high morphological diversity with two main colony morphologies. In one hand, we have whip corals, which consist on a single unbranched stem that can grow straight, contorted, and spiral. And in the other hand, they can present a branching pattern that can resemble to a bottle brush, bush-like, fan-shaped, feather-like. They are found worldwide from polar to tropical regions and from shallow water to abyssal depths. And they have a high economical value, being heavily threatened by illegal harvesting for black coral precious jewelry. They are suspension feeders considered to feed in zooplankton, and some species have been observed contracting their polyps during the day and expanding them at night, suggesting that they may feed only at night. In addition, they present also variable polyps and mouth size. Like here, this is a branched coral who has smaller polyps and mouth than whip corals. However, information on their feeding behavior is very scarce, and only a few studies in trophic ecology have included them, but the majority have focused on deep sea taxa. Because of this knowledge gap, I wanted to assess the potential trophic diversity among shallow water black coral species. In order to do this, 3 specific objectives were set at the beginning of this project. First, since plankton is composed of many different taxonomic groups and sizes, I wanted to describe the main groups found among the plankton samples within the studied area. Second, I wanted to characterize the diet of black corals by identifying the relative importance of the main potential food sources. And finally, I wanted to explore possible differences in black corals' resource use between dry and rainy season and between day and night. To do this, stable isotope analysis is a very widely used tool, which is based on the principle you are what you eat plus a few per mile. This means that the isotopic composition of a consumer will reflect that of its prey. And this is illustrated here in this example. Imagine the diet of a coral, a consumer, will be composed of zooplankton and benthic particulate organic matter, or BPOM. And these preys have a specific isotopic composition, for instance, nitrogen 14 and 15. So the isotopic composition of the consumer, the coral, will be a mixed composition of these 2 preys. However, it is important to know that due to differences in chemical and biological processes between the heavy isotope and the light isotope, it will cause a slight enrichment in the consumer relative to its prey. And the benefit of this is that this can be measured. And it's normally expressed in delta notation, and this represent measurement is not a raw value, which is formulated here in this equation. It is expressed in per mil units, because otherwise absolute abundances are very small. Now, these isotope values are normally presented in the isospace here in this graph, where we have on the horizontal axis delta 13C, which represent a continuum of resource use, the use of different prey items, and in the vertical axis we have delta 15N, that represent a robust indicator of trophic position. So here another example. Imagine starting from a primary producers like phytoplankton, will have a delta 15N of around 3 per mile in a marine system. Then accounting for this slight enrichment, the next one, a primary consumer, like zooplankton, will have a delta 15N value of 5 per mile. And finally, a secondary consumer, like a coral, will be around 7 per mile. So each of these steps represents one trophic level. In addition, sulfur isotopes are also used and are very useful to discriminate between food sources. And in a marine system, they also help to discriminate between the organisms that rely on the sulfur source from the water column, that will have a value of around 20 per mile, to those that rely on the sulfur source from the sediment, that it's normally around less than 10 per mile. The black coral bed I studied was located in the northern pass of the Great Reef of Toliara in Madagascar, where all black corals are found between 10 and 30 meters. This area is characterized by two distinct seasons, where I sampled my corals. And here in this scheme, I'm only representing the sampling design for the dry season, but it was exactly the same for the rainy season. For consumers, a total of 133 black corals were sampled that represented 6 branched coral species and 6 whip coral species. For consumers, for food sources, BPOM and plankton was sampled, and some plankton was sampled at day and night, and it was sieved to obtain 6 different size fractions, from 63 to more than 2,000 micrometers. In addition, plankton samples were divided in two, one with the purpose to stable isotope analysis, and the other one for plankton taxonomic identification. Now into results and discussion. Plankton taxonomic composition varied among the different size fractions. In total, 25 taxonomic groups were identified. Here I'm just representing some taxa of the groups. And here we observe an interesting pattern where the largest size fraction here is composed only by X and euphaseeds. Intermediate size fractions are dominated by copepods that become less abundant with decreasing of the size fraction to completely disappear in this one, the smallest size fractions, that was completely dominated by detritus, which one part was mainly fecal pellets. For plankton isotopic composition, here in this carbon-nitrogen biplot, I'm representing with the dots the individual isotope values of the different size fractions, where I fitted standard ellipses to observe patterns, and these ellipses are 40% of the confidence interval of these values. So as you can see, the smallest size fraction, 63 and between 63 and 125 micrometers, was the only one significant different to the rest of the size fractions. Now for sulfur values, we observe an isotopic overlap among all the size fractions where no discrimination was found, and we see also that all the values are either close to 20 or at least they're never less than 10 per mile, indicating that these organisms rely on the sulfur source from the water column. This is representing the dry season, and as I told you, I have also tested the rainy season, but very similar patterns were found as well. Now, I wonder, why do I observe different patterns between plankton isotopic data and plankton taxonomic data? So one possible reason is because here the intermediate size fractions were composed of many different taxonomic groups, which yielded in a mixed isotopic composition with very similar carbon, nitrogen, and sulfur values. This is a limitation of stable isotope analysis, where in many other research in pelagic ecosystems have found an absence of isotopic discriminations between size fractions in plankton. Here I'm representing the same values, but I draw the ellipses for plankton samples collected at day and night, and you can see also that they were different. There were significant differences between these two. Now, to put everything into context, we have in one side food sources, which are represented here in this carbon and nitrogen biplot for the dry season, but again, the same similar patterns were found for the rainy season. And we have first detritus, which is composed of the smallest size fraction, that one that was different from the rest. Then we have plankton, night and day plankton, that were different. And it's the combination of all those size fractions that yielded similar isotope values. And finally, we have, with the highest values, BPOM. And in the other side, we have our consumers, black corals, where we can observe an interesting pattern. We have, in a lower trophic level, branched corals, and in a higher trophic level, all the whip corals. Now, to estimate the relative proportion of each of the food sources to each of the black coral species, I used this data to build a model, mixing model, which is called Stable Isotope Analysis in R, or SIRE. And here what you can see is for the black coral species, from numbers 1 to 5, we have whip corals, and 6 and 7 are the branched corals. The box plots represent from the darkest color to the lightest color, the 50, 75, and 95 credibility intervals, which corresponds to confidence intervals, but in a Bayesian context where these models are built. And what we observe is that plankton was an important food source for all the corals, where no differences were found between day plankton and night plankton for whip corals, although we observed that branched corals rely more on day plankton. In addition, detritus was also, the model estimated that detritus was also an important food source for branched corals. Now, in conclusion, even though discrepancies were found between plankton taxonomic data and plankton isotopic data, the combination of these two techniques helped to aid interpretation of the patterns observed. Stable isotope analysis identified two trophic levels with an interspecific isotopic variability between the two colony morphologies. No substantial temporal variation was observed between seasons, and even though day and night plankton were significantly different, for whip corals there was not a preference for one or the other. The models also estimated that the diet of black corals varied. And to finalize, as hypothesized, colonial morphology drives variation in the feeding behavior of black corals, and which also suggests that polyps and mouth size may play an important role in trophic ecology of shallow water black corals of Madagascar. I would like also, since I think I have some time, to ask you if you could raise your hand if you ever heard of black corals before. Okay, that's what I was actually expected. So hopefully you learn a little bit today about them. And I would like to thank you, my supervisor and promoter, Gilles Lepoix and Nicolas Esturaro, without whom this work wouldn't have been possible. Thank you very much for your attention.